Ma'am, please start introduction.
Good afternoon, everyone. I again welcome you in the afternoon session of day 15 of this very 21 days training program. In this session, we have Dr. Ravikant Agarwal sir with us, who is doing an excellent work in the field of food safety. Now, I would like to formally introduce him. Dr. Ravikant Agarwal has passed BVSc and AH from CVSc and AH GB Pant University of Agriculture and Technology, Pantnagar in 2001, and then MVSc and PhD in Veterinary Microbiology from ICAR IVRI, Izatnagar. He has more than 13 years of experience in teaching and research in the field of pathogenic microbiology, microbial biotechnology, and food safety. He started his career as research associate in AINPHS program at IVRI Izatnagar itself. He served as lecturer in Department of Biotechnology at Modi Institute of Technology and Science, Lakshmangarh, Rajasthan. He joined College of Animal Biotechnology, Garhwasu, Ludhiana in 2008 as scientist and served there till 2012. Then he shifted to ICR IVRI as senior scientist in 2012. Since then, he is looking after food microbiology laboratory in LPT division ICR IVRI Izatnagar. His research interests include rapid detection methods for microbial pathogens, antimicrobial resistance, alternative microbials, foodborne pathogens, and food safety. He has guided 4 PhD and 5 MBSc students till now. He has published more than 90 research articles in journals of national and international repute. Dr. Agarwal has authored 5 books in the area of microbiology and biotechnology. He has been associated as core faculty member in the training organized by Bioinformatics Center IVRI Izatnagar and Entrepreneurship Development Program organized by Agri-Business Incubator ICR IVRI Izatnagar. Thank you, sir. We are lucky to have you, sir. Over to you. I request you to please deliver the talk. Thank you, sir.
Okay, thank you, Dr. Gauri, for nice introduction. I am switching to my presentation. Am I audible to all of you?
Yes, sir. Please carry on, sir.
Okay. So, good afternoon to all of you. I am Dr. Ravikant Agrawal, and as Gauri told, that basically by education I am a microbiologist, or more specifically a bacteriologist, and I am looking after this food microbiology laboratory at division of LPT. And this is the topic which is given to me, which is microbial safety of livestock products. So I have divided this into 3 parts. One is overview on microbiology of livestock products. So just we will have an overview that what are various microbes which are responsible for causing foodborne disease, or you can say spoilage. Then some more elaborative way, I will be covering methods for microbial quality assurance. So second aspect what I will have is methods for microbial quality assurance, and in this we will take conventional again as well as rapid methods, and some part which I will touch is food laws in Indian context, various agencies which are regulating this food quality in our country. So starting with the introduction, as we all know, India has immense livestock wealth. We are number 1 in livestock population, which is 535.82 million. And as per 20th livestock census, which is recently been published, then India produce about 8.6 million tons of meat, and 114.38 produces 114.38 billion eggs as per annual report of DASD 2020-21. We are fifth in meat and third in egg production. We are largest producer of buffalo meat and second largest producer of goat meat. And livestock sector as such contributes to about 26% to the agricultural GDP and 4.5% to the national GDP, or you can say total GDP. The current processing levels in poultry sector, they are about 6% of the poultry we are processing, while in meat it is about 21%. India exported meat and poultry products worth around 5 billion USD, and this is little bit old data, 2014-15. But out of that, major portion, that is 4.8 billion USD, was buffalo meat, what we call black gold. Other export items, they included egg powder, sheep and goat meat. It means you can say eggs, poultry products, and other meat products, or you can say other meat, but it accounted only for 0.2 billion. It means our contribution to the international market is very, very low. Major export destinations, they include Vietnam, Malaysia, Egypt, Thailand, and Saudi Arabia. It means we didn't exported our meat to the developed world. Poultry is the highly vertically integrated industry in India. So one of the sector which is very much competent in India is poultry sector. And that is why Government of India has taken steps for modernization of municipal abattoir to provide safe and hygienic meat to the consumers. So there are certain issues in the livestock products that where we are lacking, and that's why our export is very, very low. If we talk about milk, so this is a happy situation that we are number one in milk production, and our milk production has reached to 187.7 million metric ton in 2018-19, which was very, very low of 17 million metric ton in 50-51. And per capita availability is 406 gram per day as per DHT report, while world average is quite low of only 302 gram. That's why we are first in milk production, but also we are number one in milk consumption. About half to two-thirds of the milk which is produced comes from small farmers from unorganized sector. Contribution of females is tremendous. But again, the same issue as with milk, as with meat and egg, that India's global participation in milk trade is negligible. And if we talk of milk products, if we talk of meat product, poultry product, exports of all these products is very low due to poor quality. Now, what are various quality attributes what we should comply if we want to increase our export? Then they include sensory attributes, nutritional attributes, processing attributes, and most important one is hygienic attributes and legal issues, under which what I am discussing today is microbial quality. Other hygienic attributes include adulteration, additives, residues. So to increase the export potential, along with the microbial quality, we should also improve upon that our product should not contain additives, or if they contain permitted additives, they should not be in higher amount. Then residues, including antibiotic residues, pesticide residues, then heavy metal contamination, they should not be there. And another very important issue is adulteration, that, or you can say, misrepresentation. So coming to food safety, it is very important global issue due to, as we all know, international trade and public health implications. Despite advances in hygiene, consumer knowledge, food treatment and processing, foodborne disease, they still represent a significant threat to the public health worldwide. The term foodborne disease in general, it covers all those illnesses which are acquired through consumption of contaminated food. So in response to the increasing number of these foodborne illnesses, foodborne diseases, government all over the world, they are intensifying their efforts to improve food safety. So now all governments all over the world, they are putting more and more efforts in the area of food safety. Why it is so? The reason was previously mainly in the developing countries, the program, they were more and more focused to provide calorie needs and minimize hunger. But now, since we have attained the self-sufficiency. So now government is putting more pressure or more emphasis on the safety aspects. Coming to microbial food safety, so one of the reason for this concern of microbial food safety is growing international trade, migration and travel. It has increased the spread of dangerous pathogens and contaminants in foods. Now, just few days back or few months back, you must have heard about this avian influenza. So many outbreak in Himachal Pradesh, in even Uttar Pradesh, they were reported in South Himachal, Uttar Pradesh. Due to globalization of food marketing and distribution, both accidentally or intentionally contaminated food can affect health of people in many countries at the same time. As we all know, world is today interconnected and interdependent. So if there is a local foodborne outbreak occurring in some area, in some part of the globe, it can become a potential threat to the entire globe. And this has happened several times. In 1991, cholera started from the coast of Peru. It rapidly spread across Latin America. It led to 4 lakh reported cases and more than 4,000 deaths. And these 4,000 deaths, they've spread it in several countries. So this is the potential of now, due to this interconnected connectivity and movement, migration, travel, if there is a local foodborne disease outbreak or local disease outbreak, this can spread to entire globe. And this we have experienced very well, although it is not a foodborne disease in terms of SARS-CoV-2. This we have experienced and we are still experiencing. The identification of one single contaminant, apart from this, if even a single contaminant is identified in your food consignment, then this can lead to heavy economic losses in terms of production. Then after that, there will be trade embargoes and there will be damage to the tourist industry. And again, the example of this is in early 2008, an outbreak of avian influenza occurred in Bangalore, and this led to the import ban on Indian poultry products to the Middle East. As I told, these are the countries where our meat products, they are mainly exported. So the Middle East imparted ban to the poultry products, and this has led to the losses of thousands of hundreds to thousands of USD to the Indian economy. The term foodborne disease, which is sometimes we trivially called food poisoning, it covers illnesses which are acquired through consumption of contaminated food, and this includes foodborne infections as well as intoxication. Globally, WHO has estimated that about 3 to 5 billion episodes of diarrhea and more than 1.8 million deaths occur in children below 5 years of age, and a significant proportion of these deaths they occur due to consumption of food, mainly food of animal origin, and that too which is contaminated with microbial pathogens or toxins. According to CDC, an estimated 76 million cases of FBD or foodborne disease they report annually in United States. Now, United States has very well implemented the food safety issues. They have implemented their food safety is very, very good, but still 76 million cases of foodborne disease they are reported in United States with approximately 5,000 deaths. And this foodborne disease occurrence, it lead to a heavy economic loss of about 9.3 to 12.9 billion. This is the cost patent for the human disease. And the major pathogens which are involved include Salmonella, Campylobacter, E. coli, O157S7, then Listeria monocytogenes, Staph aureus, and Clostridium perfringens, and these all are foodborne. Now, due to increasing number of these foodborne illnesses, as I told, governments all over the world, all over the globe, they are intensifying their efforts to improve food safety. Now, this was the worst scenario. If we talk about Indian scenario, developing countries, they bear the brunt of the problem. The reason is there is a wide range of foodborne pathogens, occurrence of wide range of foodborne pathogens. There are favorable conditions for them, and these favorable conditions are mainly due to gross unhygienic practices. The burden of infectious disease continues to be a major constraint in sustained agricultural development efforts and food security. It has been estimated that about 5 lakh children below 5 years of age, they die each year due to diarrhea. And as we know, diarrhea usually occurs due to foodborne infections. Now, these figures, they jeopardize international and India's own development efforts, and including the achievement of millennium development goals. Now, at present in our country, reporting and surveillance of foodborne disease, it is grossly neglected. Therefore, the exact extent of the problem, as well as uh, in developing countries, including India, it has not been fully understood. Now, we all experience this whenever there is any diarrhea, there is any vomiting, then usually these diseases, they are self-limiting, and that's why we never go for identification of the organism or identification of the food which was implicated in causing the foodborne disease. So we never go for that, and that's why it happens that never it is reported. So most of the foodborne disease outbreaks, most of the foodborne disease cases, they go unreported. So the actual scenario of foodborne disease can emerge only with proper emphasis on foodborne disease surveillance, including identification of the foodborne pathogen involved. And this there is requirement to establish a national foodborne disease surveillance system. Now, coming to etiology of these foodborne disease, till date, more than 250 different foodborne disease, they have been described, and bacteria are responsible for two-thirds of them. And you can classify various foodborne disease into, depending on the etiology, we can classify them as bacterial foodborne infections, mycotic foodborne infections, viral, parasitic, and protozoal foodborne infections. So since we will not be going into detail of each of these, so to name them, mainly Vibrio, then Salmonella, E. coli, then Campylobacter, Listeria, even Brucella and tuberculosis, they are also milk-borne infections, as both these two organisms may come out in milk. And if animal is infected, and if somebody is consuming non-pasteurized milk, then there are chances of getting these infections. Among mycotic, they mainly include Candida, Sporothrix, and Vengiella. Viral, we include hepatitis A and D, Rota, Norwalk virus, or more commonly we call them noroviruses, and poliomyelitis virus. Among parasitic, we have hydatidosis, teniasis, anisakiasis, trichinosis. Among protozoal, we include cryptosporidiasis, toxoplasmosis, sarcocystosis, and cyclosporiasis. So these are the various infections which we can get from foods. Now, we can also classify them into foodborne infections, intoxication, and toxico-infections. If we want to differentiate them, usually infections, they are caused by bacteria, virus, as I told, bacteria, virus, parasite, and protozoa, while if we are calling them intoxication, they are caused by preformed toxins. So usually infections, they take time in establishment. They have some incubation period. So their incubation period is hours to days, while in case of intoxication, you will get response in minutes to hours. Primary symptom in case of infection will be diarrhea, while primary symptoms in case of intoxication will be vomiting, although vomiting may be followed by diarrhea. Similarly, in case of infection, diarrhea may be followed by vomiting. Infections are communicable, while intoxications are non-communicables. So these are the basic differences between infections and intoxication. Now, another aspect that is very, very important in microbial safety of food is food spoilage. Now, food production, as you know, food production occurs at specific areas, specific time period of the year, so you need to store them. But the problem starts as soon as you are going to store the food, there will be spoilage. Now, one of the reasons, there are several reasons for spoilage, but the most important factor is microbial food spoilage. So you can call them food decay, food decomposition, food spoilage, and there can be microbes are one of the important factors. So among microbes, I told bacteria, viruses. For foodborne disease, I told bacteria, viruses, fungi, protozoa, parasite. But for food spoilage, bacteria and fungi, they are mainly responsible for food spoilage. So major causes of food spoilage, there, as I told, there are several causes, but growth and activity of microorganism is one of the important cause of food spoilage. So why food spoilage will occur? The food spoilage will occur because microbes, they will get entry into the food. Either it may be egg, either it may be meat, either it may be milk. Because as we know, when milk, if we are taking producing meat from a healthy animal, it is sterile. But due to handling, it gets contaminated. Similarly, when milk is produced inside the udder, it is sterile. But as soon as it comes into the teat canal, it gets contaminated. So, due to handling, so major sources of microorganisms in food are soil and water, intestinal tract, food handlers, utensils, animals' hides and skins, air and dust, and plant products. So, once microorganism will get entry, what will happen? There will be several steps in microbial food spoilage, which we classify them into intrinsic factors, extrinsic factor. As microbe will be introduced into food, food environment should be favorable for them. So the various factors which are associated with the food itself, they are called as intrinsic factor. Similarly, where we are storing our food, so a proper temperature, moisture, gaseous environment, if we are providing, they will flourish. So we classify them into extrinsic factor. So these intrinsic factor, extrinsic factor, they decide at what pace, what speed these microbes they will grow, and they will cause spoilage. So how we can visualize, it is not always necessary that if your food is spoiled, then there will be visible change. But most often we see some visible changes, like there will be visible growth, as I have showed in some photographs, that clearly we can see that some fungus is growing. Then oxidation of pigmented products can occur. This can change color, and even there are certain microbes which produces color. There are fungi, candida. Then we have bacteria which produces color. So that can change the color of your product. Then breakdown of tissues and degradative enzymes can change texture. Production of dextran in sheer number can produce slime. There are so many microbes which produce dextran and other amorphous material outside their cell, so which produces sliminess. Then water can be released, that is purging can occur, gas production can be there. So these are the visible signs of spoilage which we can observe in a food. But as I told, it is not always necessary that a visible sign is present to consider a food is spoiled. Like if let us say if some Staph aureus came to any food product due to handling, and instead of and it was toxigenic, as you know that Staph aureus they produces enterotoxins, and these enterotoxins, they are heat stable. Then although it may be possible that the bacteria may not have grown too much to produce spoilage, but it has produced sufficient amount of toxins to cause foodborne intoxication. So there are so many type of microbes. Some of them, they are listed here, some common bacteria, fungi. So these are responsible for food spoilage. Now we talked about foodborne disease, that microbes, they are responsible for foodborne disease. We talked that microbes, they are responsible for food spoilage. So Food Safety and Standard Authority of India, which nowadays is the primary agency which is governing the quality of food and food products in India, they have given certain guidelines. They have notified in the form of a gazette some microbial standards for various food products. So since we are concerned only with the livestock products, so I have taken those tables here and I'm showing. So we can, as we talk about microbes which are responsible for spoilage, and we talked about microbes which are responsible for foodborne disease. So similarly, we can classify these criteria into two categories. One we are calling here process hygiene criteria. So these criteria, they define that how you have processed your product. Let us say if I take example of meat, then how you have slaughtered animal, how you have handled it to reach it to the consumer. So these so that the number of microbes should not be too many to cause spoilage of food. So these all come under process hygiene criteria. So in process hygiene criteria, we see total plate count, or here we are writing aerobic plate count, total plate count, standard plate count, all one and all the same things. So aerobic plate count, yeast and mold count, Escherichia coli, it mainly indicates the fecal contamination of your product, and Staphylococcus aureus, which is coagulase positive. So in process hygiene criteria in meat and meat product, we measure these four important test we perform, that is, we perform tests for aerobic plate count, yeast and mold count, E. coli count, and Staphylococcus aureus count. And Food Safety and Standard Authority of India, they have classified various meat and meat products into various categories. So for each category, these four counts, they have been given. And here N is the number of sample which we need to take for evaluation. C is the number which can which can show limit between M and M. M is the minimal limit, and capital M is the largest limit. And C is that number which can show, which can yield result between this and this. It means if, let us say, for fresh meat and chilled meat, 5 is the number of sample which we need to collect. C is 3. That is, 3 number of sample can have number between 1 into 10 to the 6 to 5 into 10 to the 6. None of the samples should exceed capital M. And if number of C, it means if more than 3 samples, they are exceeding small m, then also your lot or your consignment will be rejected. So similarly for milk and milk product, we have process hygiene criteria. Here again, we are having aerobic plate count, coliform count. Excuse me. Just a moment, please. So again, we have aerobic plate count, coliform count, Staphylococcus aureus, yeast and mold, and E. coli. So again, here, instead of 4, we have 5 different counts. So again, FSCI has divided milk and milk product into various categories. 14 categories are there. Similarly, as we talked about foodborne disease, so FSCI has separately given microbiological standard for meat and meat product in terms of food safety criteria. So as I told, there are so many bacteria which are responsible for causing foodborne disease. So we mainly have Salmonella, Listeria monocytogenes, sulfite-reducing Clostridia, Clostridium botulinum, and Campylobacter species for meat and meat products. And as I was telling there, you can say that sampling for food hygiene criteria, we have 3-class plan, while for food safety criteria, we usually follow 2-class plan, where we only have yes and no. But mainly for Salmonella and Listeria, we follow 2-class plan, where sample number is mostly 5. It means for each lot, you need to have 5 number of sample you need to take. And here C is 0. It means none of the sample is allowed to cross the prescribed limit. And for at least Salmonella and Listeria, they should be completely absent in most of the meat and meat products. Or you can say from most of the food and food products, Salmonella and Listeria monocytogenes, they should be completely absent. But for sulfite-reducing Clostridia, Clostridium botulinum, Campylobacter, we have different numbers. And here we are following, although we are talking about food safety criteria, still we have some, still we have 3-class plan. It means we have number of samples, we have certain number of sample, like here it is 2 in case of cured pickled meat. In case of sulfite-reducing Clostridia, 2 number of sample can exceed 500 limit. It can go up to 500 to 5,000, but not more than 2 sample should show more than 500 number of sulfite-reducing Clostridia, and none of the samples should exceed 5,000 limit. Similarly, for milk and milk products, we have food safety criteria, and here we have Salmonella, Listeria monocytogenes. As I told, again, for Salmonella and Listeria monocytogenes, strictly none of the sample should be exhibiting Salmonella or Listeria monocytogenes. Even if one organism is detected, your food is liable to be rejected. And in milk, we additionally have Bacillus cereus, sulfite-reducing Clostridia is same. And here we have Enterobacter sakazaki, which is renamed as Chronobacter. So now we call it Chronobacter sakazaki. So again, in 14 categories, food safety criteria is also there. So till now, I covered my first part, that is general introduction to food microbiology. Now we talked about spoilage, we talked about foodborne disease, we talked about food process hygiene criteria, we talked about food safety criteria. But for following all this, I told you that since FSCI is the primary governing body, which is meant for ensuring food safety in India now from 2006 onwards, so they have laid certain guidelines, they have notified various standards in terms of not only in terms of microbiology, since this is related to my topic is related to microbiology, in all terms, in terms of heavy metals, in terms of pesticide residues, in terms of antibiotic residues. So they have given criteria that your food should not exceed this level. Since my topic is microbiology, I am taking only that part. So if we want to, so now if I have to ensure quality of a particular product, either it is a milk, meat, or egg, then how to do this? So since there are two type of criteria, we have process hygiene criteria, we have food safety criteria. So I will be having two requirement: one, to measure the total microbial load in a particular food product, and additionally I need to let us say in case of salmonella and listeria, I need to tell that this food is either having salmonella or listeria monocytogenes or not. So specific pathogen or their toxin, they should be absent. So we need to have something for pathogen or toxin detection. And similarly, we need to, since we need to measure, there are certain limits which are prescribed by FSCI. We need to have methods for measuring the total microbial load. So traditional method, now I will talk about some traditional and molecular method, or you can say not molecular, modern methods, rather I should say. So traditionally, whatever methods we are doing for microbiology of food, they pose several challenges, like growth of fastidious or fastidious organism. It is slow. Some organisms, they are non-culturable. Then maintenance of viability. Let us say if some food is produced, let us say 50-60 kilometer away from the laboratory, then transportation, the maintenance of the viability. Then if you are let us say I want to do I want to detect presence of salmonella in a food sample, then I have to culture that food. I have to culture food sample in the laboratory. So there are chances of hazard if technical expertise is not proper. Additionally, you need technical expertise, and these all procedure, they are costly. If we talk about molecular method, or you can say modern methods, then they are claimed to be faster and simpler, more sensitive, specific, better adapted to instrumentation, and also possibility of epidemiologically important identification of epidemiologically important strains is also possible. Now we are seeing in COVID scenario also, you must be going through alpha, beta, gamma, delta variants. So these are all possible. These are not possible by conventional method. Let us say if I am putting sample of my throat swab into some cell line, then it will tell me only that either COVID-19 or SARS-CoV-2 is present or not. It cannot tell that this SARS-CoV-2 is how it is different to the previous SARS-CoV-2. So this is the advantage of molecular method, that epidemiological surveillance can be done, as we are seeing in case of SARS-CoV-2. In real time, scientists, they are monitoring what are the changes that virus is acquiring over a period of time. So nowadays, recent research focus in terms of food microbiology, in terms of food safety, is to develop rapid methods for quantification of microbial load, which is our requirement in terms of food safety, microbial food safety, rapid methods for detection of foodborne pathogens or their toxins. Additional to this, there are work is going on rapid methods for species identification. As I told that certain legal issues are there in terms of adulteration. Then for residue analysis, like pesticide residues, veterinary drug residues, heavy metals, additives, contaminants, and as well as intelligent packaging applications. So these are the recent research focus in terms of food safety. So I will quickly go through what are various traditional methods and rapid methods for enumeration, that is quantification of microbes. So first is, as we are doing routinely in our laboratory, we go for plate count, and there are various plate count method. Excuse me. Hello. Hello. We have pour plate method, spread plate method, drop plate method. These are the method which are routinely doing in our laboratory. We used to collect sample, make homogenize it, make serial dilution, and we go for plating in the triplicate, then multiplying the count with dilution factor. We can easily calculate what is the number. Membrane filtration method, they are usually used for liquid foods. Apart from this, especially in case of milk, we are going for various dye reduction method. Most commonly used method is MBRT, methylene blue reduction test, where blue color of the dye is changed to leucomethylene blue, and more number of organisms, it means there will be early conversion of this methylene blue into white color, that is leucomethylene blue. So on the basis of that, we correlate that what is the quality of the milk. Beside that, we can also use this TTC dye, rizazoline dye. They can also be used. Similarly, ATP photometry can also be used, and this type of system requires the use of luciferin luciferase system. So since we know that ATP is the energy currency, and every bacterial cell is having 1 femtogram of ATP. So according so counting of the photons is done by using luciferin luciferase system in ATP photometry. Similarly, direct epifluorescent filter technique, this can also be utilized here. This is very much similar to membrane filtration technique, but here we are also using one DNA RNA staining agent, that is acridine orange. And the basic concept of this is that if in a cell, if in a cell there is more amount of DNA, then acridine orange after binding will fluoresce green. If there is more RNA, it will fluoresce orange. So if a cell is live, it means it is actively transcribing its DNA. So it will be having more amount of RNA and it will be fluorescing orange. While in case of dead cell, there will be no RNA, so it will be fluorescing green. Similarly, electrical impedance-based methods are also there, and vectometer is one example. So, but nowadays it is obsolete technique. Coming to pathogen detection method, conventional method. So, as we all know that once we are going for any pathogen detection method, it is a time-consuming process. You need to have a sample homogenized, then you need to go for enrichment, then secondary enrichment or selective enrichment, then you need to go for plating on selective media. And after that, you need to go for various biochemical tests and followed by serotyping. So it is a 5-step process: culture, or you can say isolation, then macroscopic morphology, microscopic morphology by staining, then biochemical testing and serotyping. So this whole procedure takes about 7 to 10 days. So there is a need to develop rapid test method, rapid diagnostic method. So what we should call a rapid diagnostic test? A rapid diagnostic test is a diagnostic test that should be quick and easy to perform, and should provide same day result, more specifically in 2 hours and typically in 20 minutes. So to be frankly speaking, whatever molecular test even we are using, ideally it is not a rapid method, because it does not deliver result in 20 minutes or 2 hours. It also takes about morning to evening time. So turnaround time is about 6 to 8 hours. So WHO recommends that any ideal diagnostic test should have or should be having a assured criteria. What is the meaning of this assured? It should be affordable, sensitive, specific, user-friendly, robust, equipment-free, and D stands for deliverable to the end user. So coming to pathogen detection rapid methods, I have divided these into 3 categories: automated growth-based technologies, then various molecular method, and cellular component-based technologies. So these automated growth-based technologies, they are nothing but these are the systems where we are having various machines, and these machines, these instruments, they are devices to incubate, agitate, and monitor culture bottles for microbial growth. But they are providing ideal environment for any sample, ideal environment for incubation, and they also have advanced culture media. There are three, at least 2 are the major players. One is Biomiriu, and another one is Becton Dickinson. These are the 2 major players in culture automated automation. That is, they are providing machines which can give you automation in the in the growth of the culture medium, and they will only tell you, they will not give you these bacteria bact alert and backtech system. These are the two systems which are provided by bioMerio and BD. And as I'm saying, they are culture automation method. That is, they will tell you that either your sample is contaminated or not. They will not tell you that which organism is involved. So this is the principle what they follow, and why they are more efficient than visual detection what we usually follow in a microbiology laboratory. They on the These are the pre-prepared bottles they provide, and at the bottom of the prepared bottle they have a gas permeable sensor. So what is the principle behind it? As soon as there will be growth of any microorganism in your food sample or any other clinical sample, CO2 will be produced, and this is a gas permeable sensor. So due to presence of CO2, this sensor will change its color from gray to yellow. So continuously light is reflected, light is incident on this sensor. So as soon as there will be change in color from dark to light yellow color, the machine will read the reflectance and immediately alert you by giving an alarm that some growth in the bottle has started. So that's why these systems, they are very, very efficient. So whatever I was telling, an LED will be giving some light, and as soon as this sensor will change its color, the reflected light will be measured by the photodiode which is available in the machine. So that's why they are very, very efficient. So similar type of bottles there, and instrument is also provided in the name of VersaTrack. This is newer instrument again from bioMerio. This is the instrument which is named as Communex technology, and this is a labeling based system. As we know that most of the time in our food we have organisms which are either environmentally stressed, nutrient depleted, or they are present in presence of growth inhibitor, or they are surviving in presence of growth inhibitor. So you can say they are stressed, damaged organism. So once we are going plating them for conventional microbiology, what will happen? They will not grow. They will not grow, and they will die. So organism was present, but our interpretation is that there is no organism in that sample. So this bioMerio has come up with a solution that if the microorganism is environmentally stressed, nutrient depleted, or present in presence of growth inhibitors, so what they are doing, they are labeling these with the help of fluorescent dyes, and these fluorescent tagged microbes, they are counted by this machine. So yes, no result, they require no interpretation in this, and the machine generate result within minutes after 24 hours of incubation. So no doubt time requirement is more, but efficiency of these type of system is very high. Coming to pathogen detection methods. So pathogen detection method, Vitec is a completely automated instrument that offers a rapid result. And not only it, nowadays you are knowing that antimicrobial resistance is another very big issue. So not only it automatically identifies the pathogen, but also it performs the antimicrobial susceptibility testing. But we should have organism in on plate for identification purpose. So these are, before that, what I was telling, they were the culture automation methods, and this is the automated pathogen identification systems, Vitec. And similarly, another system we have Phoenix, which is obtained from Becton Dickinson, and both are similar type of system which help us in identification of pathogens with turnaround time of 2 to 6 hours. And additionally, it also performs the antimicrobial susceptibility testing, which is nowadays very, very important, and that is a global issue. Another type of automated system is API, that is Analytical Profile Index, which is again original concept was developed by bioMerio, although it is nowadays available from High Media. So these are the miniaturized system for identification of the pathogen. Whatever biochemical test you need to perform, you can perform these tests on the miniaturized strips. And nowadays even softwares are available. Using those softwares, you can easily identify the pathogen following these API strips. Coming to the molecular method. So whenever we talk about molecular method, it comes to our mind as these are nucleic acid based method. But either protein based method or nucleic acid based method, they both come under the molecular method. Molecular method we can divide into, in case of nucleic acid based method, we can divide them into hybridization assays, amplification assays, also nowadays genome sequencing, which we are seeing in case of SARS-CoV-2, and also protein-based methods. So coming to nucleic acid-based methods, why they are advantageous as compared to the hybridization based methods? Because our food can have very low number of organisms. So if a number of organisms are very low, then hybridization based method may give us false negative result. So to overcome this, nucleic acid based method, they have advantage over the hybridization based method. So most common example is PCR. Everybody know this. Everybody must be doing in your laboratory. So PCR-based methods, they are quite developed, and they are three-step methods, which include annealing, which include denaturation, annealing, and extension. And finally, we will be having millions of copies of a particular DNA sequence produced. So this is on the left. I am showing you how this PCR will occur. There will be denaturation. The two strands will separate out. Then there will be annealing of the primer, and finally there will be extension in each step. So there will be exponential growth of the target gene, and finally we will be having about 28 copies of the target gene after around 30 rounds of application, and the whole process can be completed in about 2 hours. This we need to, so 2 hours in amplification will occur, then we need to follow for agarose gel electrophoresis, and we will visualize these bands, such kind of bands. On the basis of that, we can detect that either a particular pathogen is present or not. So the purpose of these slide is nowadays for any diagnostic PCR, it has been made mandatory to include an internal amplification control. So internal amplification control is required to overcome the false negatives. Now, I can simply tell you why these internal amplification controls are required. Internal amplification control means, let us say if I am having this is a, let us say PCR for Campylobacter, this is a PCR for VTEC or Virotoxinic E. coli. Then if my specific band, or you can say if my E. coli VTEC is present, then I should have 2 bands. And if in a negative sample, even I should have 1 band. So we can easily assume this in terms of we must have seen the lateral flow assays, and most common one is pregnancy diagnosis assay. So in a pregnancy diagnosis test, whenever we perform, if we are having 2 lines, then it is positive. So you can consider here, if we are having 2 bands, then it is positive for a particular pathogen. If we are having 1 line, as we are seeing in lateral flow assay in pregnancy diagnosis test, 1 line means negative test. So 1 band means negative test. So this additional band is required to overcome the problem of false negatives. So now this is a recent introduction. We cannot take PCR machine to the field condition. So even miniaturized PCR machines are available, which are of palm size only. So you can, these are battery operated. These you can easily take them to the field condition. As I told that in PCR there are certain drawbacks, that you need to have a instrument, then you need to also to go for post PCR processing in terms of gel electrophoresis. So to overcome that, real-time PCR, qPCR or kinetic PCR, it has been developed. It has advantage that amplification and amplification visualization can be simultaneously on computer. So machine will amplify the product, and using various fluorescent dye, we can observe real-time amplification, which is plotted in terms of a graph. So this is the advantage of PCR, and these systems, they are more sensitive than conventional PCR system. There are various chemistries. I'm not going into detail due to time constraint. So this Techman probe, everybody must have heard the name of Techman probe because of SARS-CoV-2. Because whatsoever diagnosis, whatsoever throat swab we were sending to the laboratory, they all were performing this Techman probe based multiplex PCR to tell either your sample is positive or not. So now these are all chemistries which we utilize in real-time PCR. As I told, for PCR, we have palm PCR. Similarly, for advancement of real-time PCR machine is this droplet distal PCR, which even have more sensitivity than the conventional real-time PCR machine. Nowadays, even not for the food system, but various viruses and bacteria for clinical infections, automated PCR systems are also available. As I told, this is PCR, real-time PCR, they all have certain limitations that you need to have a thermocycler. In real-time PCR, you need to have a real-time PCR machine. So now people are moving towards isothermal amplification method. So instead of using a PCR machine, simply in a heating block, you can perform this loop mediated isothermal amplification. We are calling them isothermal amplification method. On a constant temperature, there is no need of any cycling to have degeneration, annealing, and extension. Simply at constant temperature of 65 degrees Celsius in a water bath or in a heating block, battery operated heating block, we can go for this loop mediated isothermal amplification. Another problem with PCR was you need to go for a post PCR processing in terms of electrophoresis. So here again, we can skip that by using various dyes. So this is the comparison of PCR and LAM. PCR is a cyclic reaction. Here it is isothermal. PCR takes more than 2 hours. It will take hardly 1 hour. You need to have denaturation, annealing, extension, or polymerization. So cycling is required. Here it will occur at constant temperature of 60 to 65 degrees Celsius. Additionally, in LAMP, we do not require any electrophoresis. Simply by measuring the turbidity by turbidity meter or by using some fluorescent dye like this cybergreen dye. This is negative sample and this is positive sample. If there is any amplification, this will fluorescent green color will appear. So in visible light or under UV light, you can visualize it. So there is no need of any electrophoresis. So these are the systems on which people are working nowadays. Now, this was all about molecular detection amplification method. As I told you, hybridization methods are also there. So DNA microarray is the best example of hybridization method. But this method you cannot apply to your food samples. The reason is the process is quite cumbersome, lengthy, and also costly. But these techniques are very, very efficient, very, very extensive. If in a particular sample you need to screen thousands of pathogen at a time, then this is the ideal technique to do so. Coming to next generation sequencing or high throughput sequencing, you must be seeing nowadays data is available for COVID-19, and this COVID-19 sequencing is possible so fast because of this NGS platform, or you can say next generation sequencing platforms. And nowadays, this slide I have shown that a sequencer has reduced, as I told for palm PCR. Similarly, a sequencer has reduced to a size of a palm. And nowadays, previously, we used to wait for about a month for a sequence of 1,000 base pair. Now, the whole genome of a bacteria or virus can be sequenced within 1 week. Cost are reduced 100 times. Time is also reduced 100 times. It means if we were taking 100 days for a particular experiment previously, nowadays it can be done in 1 day. If I was spending 1 lakh rupee, now this can be done in 1,000 rupee nowadays. So nowadays you can perform. So this is the size of a sequencer nowadays, palm size, which is Oxford nanopore. Most of the sequences which are published nowadays for COVID-19, lakhs of sequences, they are available in the database, and most of them they are sequenced by this Oxford nanopore machine, Minion. Coming to protein-based methods, this we all know: agglutination method, Western blot, then Western blot, and then so to enhance ELISA, and then to enhance the sensitivity, you can also go for, nowadays people are going for immunomagnetic separation. Similarly, on the basis of protein systems, we are also having these automated systems in the name of VIDUS and MiniVIDUS. Again, it is from BioMerieux, and this is one of the system which is having many certifications, and this can be utilized for food quality control as well as for personal care testing. Coming to lateral flow assays, as I gave example of that pregnancy diagnosis step. Yeah. So as I told, for pregnancy diagnosis test, we have LFAs, lateral flow immunoassays. So people are nowadays developing them for various foodborne pathogens as well as their toxins like aflatoxins. So I'm showing this slide. This is the latest research which is going on to develop. So these assays, they are one of the simplest formats. So simply you need to put a sample and within 5 to 10 minutes. So these are actually lateral flow assay fit into the criteria of assured, which is propounded by WHO, that we should have result within 20 minutes. So here we can have result within 5 to 10 minutes. So nowadays people are even trying to combine sensitivity of the nucleic acid based method with the simplicity of the lateral flow immunoassays in the name of nucleic acid lateral flow immunoassays. Coming to cellular component based technologies, so two systems we are having. One is GLC, gas liquid chromatography based system. So this system is available in the name of GC from Sherlock company. And similarly, nowadays the latest addition is mass spectroscopy based methods, and Multitop MS is one of the leader in this. And two systems are available from BD Bruker, that is Multi Biotyper, and from Vitec, from Biomerics. So these are the two instruments which are available for pathogen detection. Nothing we have to do here. We have to simply put a bacteria on this plate, put this plate into this instrument, and put metrics on this, and then we can directly the machine will automatically tell which organism is this within 10 to 20 minutes. So turnaround time is 10 to 20 minutes. So as I told, none of the system is perfect. There are certain strengths, there are certain limitations also. So I will now, since time is moving, so I will quickly go through some of the food laws. Pre-FSI scenario was there were about there were many 8 different ministries were governing 9 different laws. So this was the basis of conceptualization of Food Safety and Standards Act in 2006. So all these 8 laws, they are covered under FSSA. So FSSA was conceptualized. The act was passed in Parliament in August 2006. Authority was established in 2008. FSSA regulation was notified in August 2011. New act was implemented on 5th August 2011, and now all food business operators in India to have a license or to get registered with the FSSAI. So the major all these are all stakeholders, research institutions, industry, farmer organization, consumer organization, government agencies, regulators. They are all stakeholders of this FSSAI. There are certain salient features, that involvement of stakeholders in decision making. There are science-based standards, improved regulatory structure, improved monitoring system, improved justice delivery, promotion of innovations, consumer empowerment, accountability, private public participation in enforcement. So this is the mechanism how we are regulating it. And now one thing what I want to tell is, if your business is about 12 lakh rupees, then simply you need to register. If it is more than 12 lakh, excepting for milk and milk product, if you are dealing with milk and milk product or meat of 100 kilo or liter per day, then you need to simply food business operator simply need to register. If it is more than that, you need to take license. So, but there are certain issues in implementation, like HR requirement and other issues. There has been tremendous progress so far, but there are certain challenges also, and certain new initiatives have been taken by FSSAI, and they have established one Netiscofen, Network of Scientific. Excuse me for 5 minutes.
There's some visit at IVRI in the LPT division, so please wait for 5 minutes, I request.
I'm sorry. As I told, there was a team from UGC. So these are few new initiative which FSCI has launched. So this was one program, Network of Scientific Cooperation for Food Safety and Applied Nutrition. So here they have identified 10 institutes, and they have given different aspects like IVRI. I say IVRI is the lead institution for biological hazards in foods. And now very recently they since as I told that FSI has notified various standards, but these standards now they are very scattered. So now what FSI is planning, that they are planning to develop commodity specific monographs in the name of Foodocopia. So this is the newest initiative. In March 2021, they have given these projects to the Netiscofen lead centers, that like IVRI is developing monographs for dairy products and analogs. So these are some of this is the photograph for launch in of this Netiscofen initiative in Delhi in December 2019. So other regulatory bodies. So I will not, since time is not will not permit me. So I will not go into detail. But there are certain other regulatory bodies also, like APIDA, then Bureau of Indian Standards, EGMARC, and then EIC, Export Inspection Council of India, which is mainly focusing on export aspects. So I will be closing my lecture here, and this is a photograph where we are seeing that we started our pathogen identification from simple biochemical tests, moved to API strips, then automated system, molecular method, then real-time PCR, from PCR to real-time PCR. Then we went for next generation sequencing, and finally we have moved now to palm scale sequencer. So now science is moving very fast, and even nowadays for pathogen detection, not only for epidemiological aspect, for pathogen detection, for antimicrobial resistance, for virulence typing of the organism, that a particular pathogen is having which virulence marker, people are following these palm size minion systems, which are from Oxford Nanopore. So I will stop here.
Thank you very much, sir. Now we will be taking question answers. And before that, I really thank you, sir. It was really informative, and you have enlightened us and make us aware regarding the new technologies with respect to food safety. Now we will be taking question answers. The stuff we have one question, that can you please elaborate about next generation sequencing, uh, IRS principle and how it is done? Its principle, I guess. Sequencing and
So
its
there,
principle and how it is done.
so in layman term, if I want to explain that, if anything you want to sequence, then you need to prepare a library of that. You need to digest the product, and depending on the system which you are using, yeah, if you are using, there are different type of chemistries which are available for sequencing also. Like ABI is using different platform, Solexa is using different platform. So depending on the what chemistry you are using, so you need to prepare that particular size. You need to digest the genome, you need to prepare library, and then you need to go for sequencing of that. So after sequencing of that, there is a whole range of pipeline you need to follow to assemble that genome. So like previously we were doing Sanger sequencing, that was a different platform, but all these next generation sequencing platform, they are entirely different. So you need to exercise a lot for assembly of those sequences. So there are pipelines which are available. The more generation of data in NGS is not a tough task, but assembling that data into a meaningful genome assembly is the most tedious task, and this requires special expertise. you need to learn a lot. You need to devote a lot of time. You need to take help from, like, I gave example of Oxford nanopore. So this Oxford nanopore, the library construction, then sequencing, hardly it will take 3 to 4 days. But the data whatever is generated, to assemble that, you need to take it will take a lot of time to assemble that data into a genome.
Thank you, sir. So we have one question, that what are the microbiological standards for export of meat and meat products?
So for export of meat and meat products, if let us say if you want to export meat to US, then you need to follow their standards. Whatever standard I have shown, they are for our country. But if you want to export meat, then you need to follow. It will depend on which country, to which country you want to export meat. So you need to fit into that criteria.
Very well addressed, sir. I guess we don't have any other question. Thank you very much, sir. Am I
Okay,
audible,
thank
sir?
you. Thank you, Dr. Gauri and other organizers for giving me opportunity to interact with participants. I am seeing participants are also very high in number. Usually
Yes,
in
sir.
ICR
It's
trainings, we used to see 20 participants, 25 participants.
more
But
than 100
here I'm seeing at
sir,
least.
as such.
Okay, okay,
But
okay.
right now we are having 66 in number, but as such we are having 100 plus.
Okay, okay, okay. So many congratulations to Dr. Sudesh, Dr. Gauri, and other organizers, because I am not knowing all of them, to have these many number of participants and giving me an opportunity to interact with them.
Thanks,
Thank you,
Dr.
sir.
Agarwal. Thank you so much.
So, participants, tomorrow we will be having Dr. Gorakhnath Slip, who will be presenting on dietary potential of Himachali, Ahadi cattle and Gaddi goat meal. And in the afternoon session, we will be having Dr. Birbal Singh from IVRI regional station itself, and he will be presenting on gut microbial evolution and engineering, a translational perspective on post and
Can I
ecological
leave,
health.
Dr. Gauri? Can we
Yeah, yeah,
leave?
sure. Yes,
Thank
sir.
you, thank you, thank you, sir.
Thank you,
Bye
sir.
bye, sir.
Thank you so much, sir. So stay safe. We will meet you tomorrow. Okay, thank you. Thank you, sir.